Now the next topic according to NCERT is the Human Genome Project, and if you read even your help books also, you will find the similar language of NCERT has been copied in those help books also, because according to the CBSE guidelines you are supposed to understand only the basics of the Human Genome Project along with the RISE Genome Project which has been added, and according to the modified syllabus of this year, in this academic session you have to cover even the RISE Genome Project. So let's quickly understand what exactly is Human Genome Project. So to begin with, first of all, what exactly is genome you have to understand. Genome is to be simplest in the definition, I can say genome is the haploid number of chromosomes of a particular individual. So that would be called as genome. For example, human genome contains 23 number of chromosomes, because we all know there are 23 pairs or 46 number of chromosomes in a diploid cell. So human genome is made up of 23 chromosomes, but it is not this much information. All the genes present on these 23 chromosomes also is covered in the topic of the Human Genome Project. So far what we have learned is that the sequence of the bases in the DNA that determines the genetic information of a given organism. In other words, we can say genetic makeup of an organism or individual lies in the DNA sequence. If you wish, you can read these things from the NCERT, and side by side you can listen to or you can have the audio of this video going on. If two individuals differ, then their DNA sequence should also be different, at least at some places. Now these assumptions have led to the desire for finding out the complete DNA sequence of the human genome. So with the establishment of genetic engineering techniques where it was possible to isolate and clone any piece of DNA, and availability of simple and fast techniques for determining the DNA sequence, a very important project of sequencing human genome was launched in the year of 1990. So we need to remember that each individual has an identity that is due to his or her genetic makeup. No two individuals are similar. We all know only monozygotic twins, because their DNA is going to be similar. So except for monozygotic twins, no two individuals will have similar DNA because they differ in their genetic makeup. Different sequences in the genetic makeup are due to difference in the nucleotide sequence of their DNA, and it was therefore this Human Genome Project started in the year 1990. Now Human Genome Project is called as a mega project. So there is a paragraph coming below which tells us why should you call it as a mega project. So it is possible because you can imagine that the magnitude and the requirement for the project if we simply define the aim of the project as follows. So if you consider, we all know human genome is said to have 10 raised to the power 9. Actually it's 3.3 10 raised to the power 9 base pair. So NCERT is simply calling it as 3 into 10 raised to the power 9 base pair. And if the cost of sequencing required is $3 per base pair. So for one base pair it is $3, the estimated cost in the beginning. Then the total estimated cost for the project would be $9 billion, because you multiply this with this. Further, if the obtained sequence were to be stored in the type form in books, now because there are 3 into 10 raised to the power 9 base pairs, and this sequence has to be remembered. So if we think of writing these things in a book or type in a book, then books, now if you think each page will carry 1,000 letters. That means the letters here stands for nitrogen bases. That means A, G, T, C. So only 4 letters are supposed to be written. So those 4 letters, if there are 1,000 letters on each page, then you need a book which carries 1,000 pages. That means a book carrying 1,000 pages, and each page will have 1,000 letters, or you can say 1,000 nucleotides. And then you need how many books of such type? 3300 such books would be required to store the information of DNA sequence from a single human cell. Remember that this experiment or this human genome project was done with the help of WBC's DNA, WBC white blood cell or leukocytes DNA. The enormous amount of data expected to be generated also necessitated the use of high-speed computational devices for data storage and retrieval as well as analysis. That means to store the DNA information, to take it out whenever possible and analyze it. The Human Genome Project was closely associated with the rapid development of a new area in biology called as bioinformatics. Just remember bioinformatics. Informatics means information. So bioinformatics is the branch, it's a branch of applied biology which deals with storing the genetic information so that the information can be used for various purposes. So now, what are the goals of the Human Genome Project? So some of the important goals of Human Genome Projects were to identify all the genes in the human DNA. So there are approximately 20,000 to 25,000 genes in the human DNA or human genome. So identify almost all of them and then determine the sequence of the 3 billion chemical base pairs that make up the human genome, and then determine the sequence, which one comes first, which one comes second, A, G, T, C, this whole sequence 3 billion times, and then store this information in the database and then improve the tool so that we can analyze these data information in a better manner and then transfer this information and technology to other sectors like medical industry, biopharmacist industries and all that, and address the ethical, legal and social issues that may arise, because there are people who are religious, they are spiritual, and they believe that such type of project is against God's creation. So there are people who will raise issues, social issues, legal issues and ethical issues. So handle all these things also.